So the first speaker is Sławomir Sowa from the National Research Institute. He is the head of the GMO laboratory, and also he's a committee of European Network of GMO Laboratories. He has been involved lots of GMO testing, validation activities. So welcome, Sławomir.
Thank you for this introduction. Thank you for the invitation. I'm really happy that after so many years we all meet together. The first global GMO conference that many of us attended has been organized 15 years ago, and since then there is some improvement in also method development, method validation, but I believe this is the problem of new genomic techniques that in fact brought us here together. I have a background also in breeding and tissue culture, plant transformation. I work in a plant breeding and acclimatization institute, so we both developed, let's say, breeding lines and do a lot of basic science that is aimed to be applied in breeding. So our institute also owns the breeding companies, and are very keen on using NGTs. Sorry. I have to say that there were a lot of information about the challenges already set by Malcolm, Alessandra, Guy yesterday. So I changed my presentation last evening. And today I realized that Raymond also placed a lot of new challenges that in fact I wanted to cover. So let me put my presentation in a bit broader perspective. So I try to look from the breeder perspective, applicant, and also official control. Because we have to understand that there is a lot of, let's say, organizations involved in the GMO detection, identification, and quantifications. Not only official control laboratories, food feed producers, plant breeders, non-GMO certification agencies, but also scientists. Yes, we didn't talk about scientists. I've been working many years ago at the Hamburg University, and we were once asked to describe our transgenic lines, yes, and then people find out that not in all cases that they really know what they are working with. So this is also a big challenge. However, it's not such a big legal problem. Our laboratory is one of the national reference laboratories that act in Poland, and I just wanted to show the picture that was, I think, shown twice already. On the picture, we see the ENGL chairman with representatives of the 24 GMO laboratories from accession countries. So I think that you all well recognize the Guy van den Ede, but you might not recognize the guy on the right side, staying on the right side. It's me, much younger, but I wanted to tell that we signed the consortium agreement just a few days before Poland joined the European Union. And this was, let's say, a big day for us because we are somehow accepted to the scientific community that deals with the GMO detection and sampling. So, but I wanted to say that the problem we are discussing today is not new. In the European Commission, the new genomic techniques have been, let's say, discussed, I think, for more than 15 years. And we've been also a part of the group that contributed to one of the first reports that was published in 2011. There was a working group established by the European Commission in 2010 to evaluate whether certain new techniques, new plant breeding techniques, constitute techniques of genetic modification, and if the resulting organisms fall within the scope of the EU GMO legislation. And you might see a lot of techniques that are listed. The CRISPR-Cas was not known, but we had the type of SDN1, SDN2, and 3 representing zinc finger nucleases. I think that it didn't change. So the same challenges remained. But there are also other techniques like synthetic genomics or synthetic biology that we'll not discuss. What I wanted to say is that new techniques become once old techniques, and the new techniques will appear. So we should not concentrate on the technology, we should concentrate on the product. And I believe that the targeted mutagenesis is a very good example. It was said already that this is a kind of an umbrella term with various techniques and different products. So in fact, we are discussing different challenges to detect, identify, and quantify of different products, not the different technologies. And we have different challenges depending on different products. Using targeted mutagenesis, we can produce various types of mutants, single nucleotide variants, insertion or deletions, gene insertion, gene duplication, translocations of different sizes, so large alteration and short alteration. And I think that with the presentations also dealing with the detecting, trying to identify of particular products of genome editing, we always have to remember that this is not does not reflect the general problems. Okay, so what was the evaluation of new plant breeding techniques identification possibility? For SDN1, identification with prior knowledge in 2011, we estimated that it's not possible. Identification without prior knowledge, also no. But of course, the methods that we used at that time, they were methods based on PCR. And even now, when we have a new PCR technology, we have the digital PCR, the limitation of the PCR methods still remain. So this did not change. There are new scientific and technical reports that were published in 2018. So the new revised technical report on new genomic techniques in 2021. There was an ANGL report on detection of food and feed plant products obtained by new mutagenesis techniques. And in 2019, and there will be a new report of the working group that was also established within the European Network of GMO Laboratories to update and give the, let's say, up-to-date science information, evidence-based information on the possibilities of the detection of food and feed plant products obtained by targeted mutagenesis and cisgenesis. We also see some kind of a terminology evolution. Yes, we had new plant breeding technologies, many techniques. Some were not discussed, let's say, deeply. Then we have new genomic techniques, and now we concentrate on targeted mutagenesis and cisgenesis. And there also terminology of cisgenesis and intragenesis evolved. But I wanted to start with the EU GMO legislation and what are the main goals. My understanding is that the main goal is to protect human and animal health and to protect the environment. And other ones are also important, but for me goes as a second. So to ensure clear labeling of GMOs placed on the market, to ensure informed consumer choice. When we ask consumers if they understand the, let's say, rules of GM food and feed labeling, I think that they have problems to accept that the products that are labeled as organic product, in fact, may contain more GMOs than the products that are labeled as GMOs. Because a very few of them really understand that the labeling is based on the ingredient, not on the whole product. And of course, ensure the traceability of GMOs placed on the market. And I think this is also a big problem and a big challenge, not only in terms of our market, but also in terms of the, let's say, food import and breeding problems. So we have to remember that the validation of GMO and identification and quantification methods is in fact a part of the GMO authorization in the EU, and that the methods are developed by the applicants. So this is the challenge for the applicants, for the GMO producers, GMO developers. So if the validation fails, no authorization of the GMO can be granted. So most of the methods that are used by the official control laboratories have been developed by the applicant. And we have, let's say, we can to some extent impact which method will be developed, which technology will be used, but in fact they can develop the methods that are based on digital PCR or the sequencing. So the challenge for the applicant is to develop a method that meets the minimum performance requirements for the analytical methods for GMO testings. And we have to remember that the current NPR are based on the PCR techniques. The ENGL and the JRC, we see that there are new technologies being applied, and new working groups for the digital PCR have been established, and also working group for the new generation sequencing have also been established within the ENGL. But the current NPRs are based on the PCR techniques. So the method provider should submit data demonstrating the positive evaluation of the detection methods. And there is a lot of parameters, but I believe specificity and trueness are the most important. So what is the challenge? The challenge is to provide evidence-based data demonstrating specificity and trueness. And this specificity should be demonstrated by similarity searches against databases. We need to create new databases. And also by experimental results from testing the method with non-target transgenic and non-transgenic material, and of course the target material. So I think this is quite a big challenge to develop a method for single nucleotide variants and short indels that may not provide such a unique sequence. And this is the problem that was highlighted by many previous speakers, also taking into account that the genomes are flexible, they are evolving, and I think that you are aware of the fact that after this presentation, your genomes will be different that when you enter the room. Yes, so it's not unique to plants. It's, let's say, occur to all the plants, the organisms. And the trueness is also a problem because for some genome-edited plants, for example, SNVs, we have to accept higher uncertainty. We have to accept higher bias. And I remember the discussion we had in 2008 during the first global GMO conferences with the colleagues from the United States. We were discussing if we should change the threshold, so moving from 0.9 to 0.1, yes, if this is technically achievable. And I remember the perspective. They also wanted to say, don't talk only about the technical aspects, but talk also about the costs, how much it will cost, and what kind of uncertainty it will bring, and what are the risks related to this 0.1. And we see this when there was a new regulation 619/2011. Yes, some methods that were validated and met the 0.9 threshold requirements did not meet the 0.1. So I think this is also nothing new, but we should remember this. The size of target and non-target changes. This is a bit, when I was a bit problematic, because when I had the previous presentations, we see that some of us would like to get rid of off-targets, and some people would like to leave off-targets as a marker for the identification of new genomic techniques. This is also a kind of a problem because Emilio mentioned that 90% of the NGT products that are in the pipeline are the type of the SDN1, and it also relates to the off targets. Most of them, there are several publications, are SDN1 type. So we have another challenge to provide evidence-based data demonstrating robustness and sensitivity of the method. So is the method targeting SNVs or short indels sufficiently robust against small modification to the testing conditions? I mean, especially in complex samples. And we, have to approach a new scale of the GMO detection. The number of GMOs, regulated according to EU legislation, will grow. And even though they will be not authorized in EU, they will be placed on the market in third countries. And it's also the same problem with the sensitivity, so the limit of detection and limit of quantification I just pointed out. The other challenge, so can new analytical technologies meet current minimum performance requirements for analytical methods of GMO testing? For example, next generation sequencings. What is the applicability? What is the practicability of the method? It's also a part of the validation. Yes, so we heard already that the equipment is not broadly used, that, let's say, some countries might not access to this very expensive, not only to buy, but also to run equipment. And even though the cost of the sequencing dropped down, using with the complex samples can be problematic. What are the challenges for control laboratories? Now, control laboratories, I could ask the question, what are the challenges for the control laboratories detecting unauthorized conventional GMOs? Yes, to what extent we are able to do this, and which methods we apply. But so we have to answer the question, is there a GM event in the sample? Which GM event is present? Is this authorized in the EU? Under which regulation? What is the quantity in case of authorized GM events? And is there a GMO that fall under the EU legislation in the sample? It's a new problem when we talk about NGTs, because we have to distinguish between mutants, natural occurring mutants, mutants that were developed within the methodologies that have history of safe use, and NGTs. And the zero tolerance policy for GMOs, both unauthorized GMOs and GMOs in seed, is really very challenging for the enforcement laboratories. Sorry. We could ask ourselves how to define it. Yes, yes. Yes, I'll finish. How to define a GMO event? And I think this is also a difficult question. Yes, if all of the targets should be used as GMOs. Okay. This has been said already, so the challenges for the screening. Detection of known genome-edited events in the context of market control. So the detection, in fact, means the identification. The current methodology for detection of unauthorized, which is based on screening strategy, cannot be applied. Let me go to the databases a bit. Because we know that genomes of plants are diverse and dynamic. And there is more than 4,000 maize varieties in the common catalog. Yes, so to cover all the variability within those even authorized for cultivation in EU varieties will be very, very challenging. I leave this case study because this is known already. But there is also a challenge to ensure breeding without GMOs, as the breeding is conducted globally. I think that we are all aware of that. Not all the methods can be easily implemented in enforcement laboratories. And to sum up, I wanted to say that the scale of problems, that the scale of challenges is growing by the growing number of NGT products, growing number of modified species, increasing problem of asymmetric authorization, the availability of certified reference materials, new methods that will appear for the GMO production or modification of plants. And this will also be a problematic for ensuring coexistence and control of organic production. So in conclusions, I'd like to maybe just add that authorization of NGT products in EU might not be possible in some cases. That the zero tolerance for unauthorized GMOs will be far more challenging for us, and that currently NGT products could only be detected by control laboratories with prior knowledge on the altered DNA sequence. So we need some adaptation of the minimal performance requirements for the detection methods, and of course we need also the legal harmonization, which organisms should be classified as GMOs. So, one of the last slide, I wanted to say that changing our mindset about the GMOs and GMO detection, I think, is the most difficult challenge. Because we don't have a common understanding on the interpretation of what is GMO event. Especially that we might have a multiple events, we might have multiple edits, stacked NGTs. This is all brings a new perspective on GMOs. Okay, and this I will leave. Yes, this is the last slide. Yes. Yes.
Sławomir, the real slides.
Yeah. No, I just wanted to say that there is a chance for change. Mutation is a change. So when the European Commission asked what should be required when reliable analytical methods that can both detect and differentiate a product cannot be provided for plants produced by targeted mutagenesis and cisgenesis, 63% of respondents considered the requirements should be adapted. And there are different types of possibilities. Okay, so.
Please,
no, it's the last one.
We have to stay in the time.
I know. I wanted to finish with this slide. Now you believe me. And this is a Latin sentence saying that tempora mutantur et nos mutamur in nihis. So the time changes and we change with the time. We see a big progress in the method development, so sequencing becomes cheaper. Computational capacity is growing. And this is up to us how we use this advancement. Thank you very much, and sorry for too long talk.